Oh, we're going to start. Welcome everyone to this Solanaceae seminar online, again this Friday. So today I'm glad to introduce Rodrigo Nicolao, and he's an agronomist from the Federal University of Santa Catarina, Brazil. And he has a master's in plant breeding from the Federal University of Pelotas. Currently, he's developing his PhD project in plant breeding at Federal University of Pelotas in partnership with Embrapa Temperate Agriculture. The research is focused on the wild potatoes from the southern cone of South America, encompassing taxonomy, genetic resources, pre-breeding, and applied uses. His research interests are agricultural research, biodiversity, climate change, crop-wide relatives, in situ and ex situ conservation, plant breeding, and potato wild relatives. So today, he's going to present and introduce us to Solanum malmeanum, a promising wild relative for potato breeding. And with that, you can start, Rodrigo, with the presentation, and thank you for being here.
Thank you. Hi, everyone. My name is Rodrigo Nicolao. First of all, I want to thank the organizers of SOL seminars online for the invitation and say that it's a pleasure to be here today telling about who am I am, and to talk a little about the work I started in 2019 during my master degree with wild potatoes, and which is currently in progress in my PhD, especially with this deer potato wild relative species of cultivated potato, Solanum malmeanum, a promising wild relative for potato breeding. Then, as I say, my name is Rodrigo. I am agronomist, agriculture and livestock technicians, master in agronomy, and currently PhD student in agronomy and plant breeding in UFPEL, with collaboration with Embrapa, Clima, Temperado. But to start this talk and with Solanum malmeanum, then I could say that Solanum malmeanum is a diploid wild potato species belonging to the Solanum section Petota in the series commersonii, currently classified into the tertiary gene pool of potatoes. This wild relative was firstly collected in Brazil, in Ijuí city, at Rio Grande do Sul state, Brazil. This specimen was collected in 1893 by Malme, and then his first classification was made in 1913 by Bitter, and it's your certificate of South American wild potato to science. Then why I choose to study Solanum malmeanum? First of all, this wild relative encloses several interesting traits to contribute for the development of new potato cultivars. And then is fundamental to gather, update, and synthesize the information on Solanum malmeanum about these desirable traits, characterize and evaluate the germplasm available, and find pathway to introgress target genes into Solanum tuberosum, broadening genetic base of the world's third most important food crop. In parallel, I would like to consider some activities which are in progress in our genetic resource lab, like the maintenance of in vitro potato germplasm, DNA extraction of wild potato accessions for molecular characterization, such as microsatellites. We are working in collaboration with Udelar from Uruguay, Dr. Paula Gallero and Dr. Pablo Esperanza. Some flow cytometry analysis, and the in vivo maintenance by tuber propagation of accessions. And then in parallel, we are campaign and helping in collaborate some experiment and fetal chambers. Example here is a project of some PhD student, Ikram Bashir, which are characterizing some wild potato accessions for heat stress. And then how to know Solanum malmeanum. For first, we review the monograph for taxonomic treatments of these tuber bearings from regional treatment as American continent, starting by bitter, passed by coral, hawks and herchins, Mendes Oliveira, Matesevach and Barbosa, and then Spooner. As well, some additional literature as BDPA, BDBTD, and so on. We attribute some criteria for updating the taxonomic information. We updated the synonyms of subspecies, check the retrieval records for Solanum commersonii malmeanum. We review some voucher specimens like gene bank pictures, as well plants under vegetation cultivation, greenhouse cultivation. To check taxonomic and geographical distribution, we check some herbarial collection according the acronyms, according to tiers, and potato gene bank collections as Embrapa, Clima, Temperado, CIP, Peru, e Udelar in Uruguay. As coordinated points were extracted from different databases. And then here we can see the geographical distribution of this wild potato, which are widely distributed in Cone Sur of South America, including Argentina, Brazil, Paraguay, and Uruguay. This wild potato can be found since from sea level up to 700 meters above sea level in a wide ecological and habitats preference of those countries as Eastern subtropical province in grassland, xerophic vegetations, in mixed forests, in which are a very presence of araucaria and lucifolia, as Pampean province in Brazil, Uruguay. And this potato relatives species flowering in a wide dayland conditions, such as from April to June, according with the hawks, as well to October to July, as reported by Spooner collaboration. As well, some authors correlation this capacity to grow in acclimatati in both cooler winter season as well in the summer in hotter conditions. Some gene bank database was consulted to assess the representativeness of Solanum malmeanum in in germplasm gene banks, following some database as followed by Embrapa, Genesis, Global Portal on Plant Genetic Resource, in USDA, and as well as aiming to compile some agronomical traits. And here is exemplified by 52 accessions, which are currently reported to be conserved in gene banks. Here in Brazil, we have 11 accessions, in Netherlands 6, in external branch North Department 8, in James Hilton Institute 2, and US National Plant Germplasm System is currently recorded by 25 accessions, which are conserved. Some agronomical traits reported in this wild species is exemplified by abiotic stresses, by frost and heat tolerance, as some authors correlated this Rosette habit forming to be generally more resistant to cold and frost acclimatized. As well, they usually want to be more resistant to combat heat conditions. By biotic stresses, some accessions were reported to be bacterial resistant, especially some phenotypes which are not occurring in Brazil. By early blight caused by pathogen Alternaria solani. Potato leaf blight is also reported 80 accession, of malmeanum to be resistant. This pathogens has been known about the Irish potato famine as in 45. And here is exemplified by some painting by Bangon. Verticillium white, some accession is reported to be resistant, as for Fusarium dry root. Some accessions is also reported to be fully resistant to potato cyst nematode, Globodera rostochinensis, but susceptible for the species pallida. And some resistant agarunal nematode is also reported. Some insect pests, this Solanum malmeanum, were reported to be interesting sources to combat Empoasca fabae, Macrocifum, Euphorbe, as Mesos persica. Some viruses also were reported to be resistant. Also paleovirus, as tobacco mosaico virus. And we compile some table from accessions which we find to be resistant or tolerant to abiotic stresses and the sources, respectively. 23 accessions were reported to be resistant to combat frost, as one for heat. Then this table summarizes the biotic stresses accessions which we find. The continuous table, verticillium white, green peach, potato aphid, and so on. But more interesting, not more interesting, but the quality traits for production and industry. This wild relative possess some specificities such as dry matter content, high content of dry matter, tuber starch, the size of starch granules, as low reducing sugar content in the tubers, protein of dry matter as amylose in starch. Especially dry matter reducing sugar is some traits which influence in potato industry, which prevent blowish during the processing of potato fries and chips. So one, as leaves are contains high levels of glycoalkaloids, which can helps to combat some pests. As here, I can introduce some brief history of taxonomic treatment of this taxon, Solanum malmeanum, starting by Bitter, when he firstly described Solanum malmeanum as mainly by an herbaceous rosette forming habit, by 20 to 30 centimeter in weight, and its leaf blade about 11 to 13 centimeter long, which leaf segment of 8 to elliptic in its white corolla. Then the main difference about the sympatical species such as Sequensis, Solanum commersonii, he pointed out that the calyx flower of Solanum malmeanum is more pointed out comparing to calyx flowers of Sequensis. From Solanum commersonii, he pointed its corolla further back in Solanum malmeanum, while in Solanum commersonii the corolla folded more folded. As the leaf traits, he concluded the number of interstitial segments pairs in the leaves, followed by coral in 62. He treats Solanum malmeanum as a form of Solanum commersonii because he believes which may be a hybrid between commersonii and Sequensis, but he characterized as a low plant age, but rarely some taller plants could be found in more open areas. From this way, they can find some plants varying for its habit from rosette to a more elongated, and its leaves up to 25 long, as well 2 or more interstitial segments between each main segment pairs. Differentiated from commersonii, he attesting the more number of interstitial segment leaflets, and the corolla flowers is always white comparing with the flowers of commersonii, which are more purple bluish. And years later, Hawkins and Hertings made some biosystematic study for potatoes of Argentina, Brazil, Paraguay, and Uruguay, including Solanum malmeanum. He treated Solanum malmeanum as a subspecies of Solanum commersonii. In treating this taxon as a rosette habit, his leaves are which decreasing in size slowly from the apex to the base, and also by its corolla, which are always white. Comparing to commersonii, the main difference for these two taxons is the size of lateral segments, leaflets, which reduces in size gradually in Solanum commersonii, while reduces in size gradually in Solanum malmeanum, whereas in Solanum commersonii, the lateral leaflet segments decreasing rapidly in size. And by the corolla color, which in Solanum malmeanum is always white, and commersonii is purple. A regional taxonomic treatment of Solanaceae, genus Solanum, in South Brazil were made by Lilian Mendes and Paulo Oliveira in 2004. He follows coral and treated this taxon in the same way of coral, and then treat Solanum malmeanum as a form. In Argentina, some flora phanerogamica argentina were made in 2005 and treated Solanum malmeanum in the same way of Solanum of Hawkins and Herting. And finally, Spooner and collaborator attributed the specific species concept while performed and published in parts a thorough taxonomic revision for all the cultivated potatoes and their wide relative for North and Central America. In 2004, South America in 2016, and for Northern South America 2019. Solanum malmeanum were treated then by specific taxon by Spooner and collaborators, treated as a habitus roseate and a tall plant, which presents its corolla always white in strelata, and its pedicel articulation about the midpoint. Another specific characters considered to differentiate these species were by leaves, also by flowers and habit, comparing especially with chacoins. In following all these previous taxonomic treatments in circumscriptions of this taxa, we made a compilation of some aligned with some accessions observations. As Solanum currently can be known as a rosette forming habit, which your stolons and tubers are predominantly white to cream, as its leaves greens, which decreases in size gradually towards the base. And especially its flowers is typically stellate and white. In its berries can produce a lot of numbers of seeds, and then some specific features of the berries which forming habitats from open pollinated plants. About a little bit about the genetic variability of some South American potato species. The early immunological studies found one shared antigen in Solanum malmeanum and commersonii, which are absent in Chacoensia, pointing these closer relationships of the first two species, where later some molecular markers were used to study the diversity, and they included Solanum tuberosum, Solanum malmeanum, and Solanum commersonii. We can see which those three molecular marker systems were efficient to separate the two species. And further, other works included these three species as can be separated by AFLP analysis. So some studies about cytogenetics cite that Solanum malmeanum possesses a chromosome number of 12 and are diploid, predominantly diploid plants, but occasionally triploid plants can occur. In diploid plants, they are producing more stainable pollen than triploids, as its pollen diameter is 23 nanometer to diploids, whereas triploids were attesting the more variable. Comparing to commersonii, they tend to be smaller than commersonii, as malmeanum also produces high pollen in quantity compared of the cultivated crop, hence the high pollen viability in two methodologies used. Some works evidences the presence of some mechanism which lead the formation of non-reduced gametes in this species, in Solanum malmeanum. And then this complexity, which was observed in the previous works, taxonomic works, which I present here, were added for the morphological recognition in Solanum malmeanum, is the occurrence of diploids and triploids cytotypes, which are represented in this figure, and with distinct morphologies, where diploids are smaller than triploids in many aspects. Then, Solanum malmeanum is a tertiary diploid wild species. And then, by controlled crossed, this wild potato were assigned as one MBN in 94 in controlled process with some selected standard EBN species. As for an efficient use of this genetic resource, here I will present some works realized in our gene bank, aiming to characterize the germplasm according its mode of reproduction, as to help to find pathways to overcome reproductive isolation, allowing hybridizations to introgress desirable traits. These two sections were presented to meet some goals, as the first characterization of hybridization, system of diploids and triploids, accessions, and the evaluation of crossability relationship of Solanum malmeanum with the related Solanum commersonii and chacoense, as with the cultivated potatoes. Some here is presented The accessions conserved at the Embrapa Clima Temperado potato gene bank, including this study, is followed by accession code, species, ploid in EBN as origin and coordinates. To accomplish some aims, we design different treatments to observe the pollen production, to see if some accession produces pollen, if the pollen is successful to fertilize another accessions. And one treatment of control to test if an accession is able to self-pollinate and self-fertilizes the flowers are one trait. Tested the apomixis and testing if an accession is able to fruit seed in seeds without fertilization of the flowers. Autogamy to test if an accession is able to set fruit and seeds when pollinated by its own pollen. And allogamy to test the female fertility and attesting the allogamy of the accessions. For crossability evaluation, we made some interspecific crosses as the allele with Solanum malmeanum diploid interploides, with commersonii diploid, Solanum chacoense diploid, and 2 cultivated potatoes, Solanum tuberosum tetraploid. Emasculation, pollen collection, and preparation as pollinations were carried out in greenhouse conditions. The absence of fruit from pollinations or from each treatment were attesting the formation of fruit without seeds and considered testimonials of existence of pre-zygotic barriers. For the proper formation of well-developed seeds was criterion used to attest the success in cross from each treatment in absence of pre-zygotic barriers, and to test if there is post-zygotic barriers, the seed were sown for a germination test. 3 to 5 pistils were also collected, 24 hours between to 62 hours after pollinations, and fixed. Then still were not evaluated, but results will be published further. Here, the greenhouses conditions. We made crossing tests in 19 and 20 and germinations in 21. Here we present the emasculation process, realizing pre-anthesis. Then pollen was collected separately for our accessions and processed according to these pictures. We put to dry in BOD chambers and then identify correctly according the accession number and conserved in a medium term as for using pollinations. Then pollinations made according each treatment in the morning, touching the stigma in the pistil with the pollen according each treatment. Further germinations assays were made by inoculating seeds in acid gibberellic for 24 hours, and then we sown in greenhouse conditions in soil. Germination are evaluated weekly up to 30 days after the sowing. Some total number of seed was determined by this formula. Cross efficiency and percentage of seed germinated was also indirectly determined. 3,000 flowers were evaluated from the 10 accessions of Solanum malmeanum, and from which 2,600 were hand pollinated. Here I need to present the results about the breeding system. In control treatments, where just the flowers were bagged and nothing were made, and we observe no fruit formation in this trait. From apple mixes, where we only emasculated plants, we just see one parthenocarpic fruit formed. As in autogamy, we made self-pollinations. We can see one self-pollinated accession, which behave as self-compatible. From 14 pollination, he formed 3 fruits, totaling 51 seeds, where only 17 germinated. Again, the 448 accession formed one parthenocarpic fruit. And here a result of the self-compatible 447 diploid accession. Figure in the left, we see the fruit formed, bearing seeds, and the plantlets germinated, which 4 weeks. In treatment of allogamy, we see which, especially the triploid plants, are female infertile when do not produce seeds when pollinated by bulked pollen, and diploid show a great percentage of germinated seeds when bulk pollinated. Some summary of breeding system behavior by Solanum commersonii. We can see by accessions followed by pollen production. Some triploid accessions do not produce pollen, where they are absent of self-parthenocarpy or apomixis, also absent. We see that one accession diploid is self-compatible, while another 7 is self-incompatible, and the 2 triploid accession were not assessed due to non-produced pollen. And while the allogamy, the triploids are infertile, as those diploids are fertile. Some crossability with Solanum commersonii. Also, we may see which they freely cross with Solanum commersonii, especially diploid accessions in reciprocal ways, but triploids usually is crossing compatible. The seed is formed, produced in reciprocal crosses with Solanum malmeanum and Solanum commersonii. We see normal with pulp seed, and the good germination of the seeds from these crosses. By interspecific crossing among Solanum malmeanum and Chacoense accessions, we can see that Solanum malmeanum especially were able to fertilize by pollen of Solanum chacoense, despite the low germination. We can see which are able to receive pollen by Solanum chacoense, while the different way Solanum malmeanum do not were able to produce germinated seeds when pollinating Solanum chacoense. The results of seeds, we can see which abnormal seeds from Solanum malmeanum and Solanum chacoense crossed, and normal and plump seed from Solanum chacoense and Solanum malmeanum, but in reduced numbers and low germination. However, we can see here the germinated plant by causability relationship in Solanum malmeanum chacoense. And here we can just evidence the cross incompatibility with Solanum tuberosum in reciprocal also. They are not producing any fruit. Here I summarize the cross efficiency, attesting the more efficiency in Solanum malmeanum according in relation to chacoense, and no efficient in Solanum tuberosum because some attributes in MBN. As well, the more average seeds per fruit when crossing Solanum malmeanum and commersonii reciprocally, and the good germination by those 1 EBN reciprocal. Despite it's not so high, but is higher than chacoense in crosses. We can conclude with this breeding study of the reproductive system, which is Solanum malmeanum is mostly allogamous species. Some self-compatible genotypes are present, pointing to promising development of inbreed lines for diploid breeding, for example. But the mechanism that allows accessions BGB 447 self-fertilizes need to be explored. Solanum malmeanum diploid accessions can freely cross with Solanum commersonii, producing normal, plump, and viable seeds. Barriers of hybridization between Solanum malmeanum and chacoense are not complete, evident the likely presence of non-reduced gametes in Solanum malmeanum. Berries set abnormal in malformed seeds. However, some of the seeds can germinate, as exemplified before in the pictures. Well, these partial successful results when crossed with the 2 ABN Solanum chacoense points to alternative pathway for introgression with bridge species allowing further advances to unlock the reproductive isolation of this promised wild relative to broaden the potato crop genetic base by the means of integrations of desirable traits. Then Solanum malmeanum can usually be differentiated from its sympatrical species, chacoense and commersonii, but ongoing studies are made in Embrapa Clima Temperado potato gene banks, where are conserved about 80 accessions of potato idol relative species, including Solanum chacoense, Solanum commersonii, and Solanum malmeanum, in vivo as well in vitro. Some characterization of this accession by morphological descriptors, morphometrics, flow cytometry, and microsatellite molecular markers, and some SNP genotyping are in progress. And the review of geographical distribution and characterization of herbaria specimens Solanum malmeanum under morphological descriptors and morphometrics are also in progress. I would like to acknowledge these great plant genetic resource lab from Embrapa, the plant breeding lab, plant tissue lab, plant molecular lab, Dr. Gustavo Heiden, Dr. Caroline Castro, Laboratorio de Evolución y Domesticación de las Plantas, de Universidade de la República, for genotype of potato wild relatives of Brazil and Uruguay, especially Dr. Paula Gaeiro and Dr. Pablo Esperanza. As International Center of Potato, G2P Soil and Horizon for Potato Breeding Training offers in 2019, utilizando la diversidad de la papa en mejoramiento genético. As for important funding, CAPES, CNPq, FAPERS, and Universidade Federal de Pelotas. Well, I'm so glad to present, and thanks.
Thank you, Rodrigo. Very nice presentation and talk. So now we are open to questions. If you have a question, you can write it in the chat, or otherwise you can type Q and I will give you the word. We have a comment of Pedro Enrique Pesi, who says, great talk, parabéns, Rodrigo.
Obrigado. Thank you, Russell, for the invitation and the organizer of SOL seminars.
Thank you. Okay, Amanda Ross has a question.
Hi, that was a really cool talk. I was just wondering if there's anything about the geographic location of that self-compatible accession you found. Like, is it on the species range margin, or is it centrally located, or is there something special about the habitat, or maybe the other sympatric species?
Sorry, I didn't understand.
That's okay. The self-compatible plant you found that sets seed,
Where is
when self-pollinated, the one self-compatible accession?
I compared the many accessions.
Yeah,
Rodrigo,
so you found one self-compatible, correct?
Yes.
I was just wondering where it is from. Is it from Argentina, Brazil,
This
Uruguay? Is it from dry land area, the forest?
These accessions are from which are they occur in a wide environments, especially in grasslands and margins of fields.
Oh, Rodrigo, I think the connection was broken. Could you repeat where is the location?
The locations is in Brazil.
Okay.
Cool, so kind of on the border, the species border.
Yes.
I think there were only a few accessions from Brazil, right?
Yes, in this map, we just put some available accession, but in further works, we are compiling and probably is increasing the number of points for distribution in these countries.
Cool, thanks. And wild tomatoes, we see a lot of the time that on this edge of the species range, self-compatibility happens. So I was just wondering if we see the similar thing in potato, especially since there are so, well, I guess, a range of self-compatible, but not a lot, mostly self-incompatible. Thanks though, really cool work.
Thank you so much.
Thank you, Amanda and Rodrigo. So then we have a question from David Duches, who asks, can you clarify if the species is classified as EBN1 or 2?
Okay, nice question. So Solanum malmeanum is classified as 1 EBN, whereas Solanum chaquense is 2 EBN.
Right. Thank you, David and Rodrigo. So then we have a comment from Gustavo Hayden. He says, congrats, Rodrigo. A lot of work done, a lot of research still to be done to advance the knowledge and applied uses of Solanum malmeanum. So, great, thanks. So Gustavo says also, Rodrigo, I suggest you to briefly talk about what you are doing now with the self-compatible offspring.
Okay. From this previous work on breeding system of Solanum malmeanum, we find one self-compatible genotype, and then we produce some seeds and fruit and tubers, which are continuous selfing. Then we are in first generation of the plants from these accessions, aiming to develop in breed line as to see which mechanisms are allowing this accession to self-fertilize. We see in last month we was analyzing the pistils with lacmoid staining, because we don't are able to see UV, but we see which pollen tube growths to ovary and may confirm the self-fertilization and self-compatibility, but much have to be done still. We continue to selfing and characterizing another accession for self-compatibility.
Great, it's a lot of work. So then we have also a question from Patrick Reeves. He says, what is the usual mechanism for introgression genes from diploid waro lectives into the tetraploid crop potato? I may have missed this in your talk.
Okay. Yeah, it's depends on crossability groups of each species are into. For example, by ploidy manipulation, we can introgress some by duplication of chromosome. We can manipulate and duplicate due to transfer to tetraploid plants, or via non-reduced gametes, also using bridge species to cross, but is mainly guided. The cross is mainly guided by the ABN scheme, which we breeders can follow these crossability groups due to guide the crosses. And this way by tetraploid breeding.
Okay. I don't know, Patrick, if it's your question is resolved or you want to add something more?
Sure. I don't know if my video is going to work. Thanks, Rodrigo. Yeah, so it sounds like it's hard, or it's as hard as it usually is with tetraploid crops when the wild relative is a diploid. I wondered if you also, or anyone in the group, knew anything about using CRISPR in tetraploids to knock out genes or genes that might be of interest.
No, I think no. I think you ask if we are using CRISPR. I think I don't understand your question. Sorry.
Oh, it's
Maybe-
about, sorry. Oh, it's about the use of CRISPR to knock out some genes in this plant. I think David Duches wants to say something with respect to that.
Yes, just to make a comment, we are using CRISPR, but are using it mostly at the diploid level. But Gabriela Massa at Argentina has successfully been working with tetraploids and CRISPR to knock out certain genes. So you might want to connect with her.
Okay, that's great. Thank you. It's really interesting.
Yes, and actually there is she gave a talk a few weeks back, and you can check it out in YouTube, in our YouTube channel, where she showed all her results. Okay, so there is a lot of nice work, congrats. So I don't know if there is any other question. Rodrigo?
No.
Not for now. Okay, so with that, we are ending this Solanaceae seminar online. Thank you again, Rodrigo, for your great talk. And next, in 2 weeks, we are back with Denise Tierman with a talk improving tomato flavor using biochemical, genomic, and breeding tools. Okay, thank you very much and see you in 2 weeks.